Thank you, Steve, and especially for sort of balancing up this idea of it's also the dad that's involved as well in some of these aspects. So we're now going to have the last speaker, and that's Caroline Relton. She's Professor of Genetics and Epigenetic Epidemiology at the University of Bristol. Her primary research interest is in the application of epidemiological approaches to improve the understanding of the role that epigenetic patterns may play in health and development. And today she's going to talk about maternal influences on offspring's epigenetic and later body composition. Thank you.
Thank you very much to the organisers for giving me the opportunity to talk about my work today, and particular thanks to the previous speakers this morning that have set the scene very well for the talk that I've prepared. So as we've heard already today, the evidence is pretty compelling that maternal overnutrition and maternal undernutrition can have adverse consequences for offspring through their effect on subsequent adiposity. And really what we're here today to discuss are the potential mechanisms underlying those observational associations and whether epigenetic mechanisms play an important role in explaining those associations. So in thinking about the epigenetic processes as a mediating mechanism or a potential mediating mechanism linking maternal over or undernutrition and offspring adiposity, we want to consider here our epigenetic modification as the mediator. And it's worth mentioning here that although the talk is very much framed around DNA methylation and epigenetic mediation, one could substitute epigenetic modification here for microRNA expression, metabolomic profiles, or other gene expression or microbiome. So the same principles exist for consideration of a number of different mediating mechanisms, but the primary interest here obviously being epigenetics. So as we've heard already, it's now increasingly straightforward to identify observational associations between a phenotype or an exposure of interest and DNA methylation. But the challenge comes in trying to decipher whether that association is causal or just a bystander effect, or is indeed reverse causation. So we have this scenario on the left here where we have an exposure that we may want to consider in relation to DNA methylation and our phenotype. So maternal obesity, offspring methylation, offspring adiposity. But which of the scenarios shown here on the right are the ones explaining the relationship that we have observed? Are we indeed seeing a case where maternal overnutrition alters offspring methylation and subsequently impacts causally on offspring phenotype? Or are we seeing a situation where the exposure is altering the offspring phenotype and methylation is consequential on childhood adiposity? Or are we seeing a situation of confounding? And my interest lies in using epidemiological approaches and population-based data to try and unpick that or to try and address that challenge. So in trying to, as I say, address that challenge, whether epigenetics is a potential mechanism mediating maternal influences on childhood adiposity, we might go about this in a stepwise fashion. So firstly, we would want to establish the association between our maternal factor and our offspring adiposity. So here we have body weight or gestational weight gain during pregnancy and offspring adiposity. As I've said, the observational evidence there is very strong. Step 2 is establishing a relationship between the same exposure and the child's methylation. Step 3, the association between the child's methylation, or the mediator in this instance, and the outcome, the child's adiposity. And then implement a variety of different causal analysis or methods to strengthen causal inference. So the work that I'll present today is based on data we've generated in the Avon longitudinal study of parents and children. For those of you who aren't aware of the study, this is a Bristol-based, UK longitudinal cohort study of 14,000 pregnant women who were recruited in the early 1990s and who themselves and their children have been followed up extensively since then. So the children are now 23, 24. Just to note, the data generated and measured throughout the lifespan of this cohort is publicly available for for anyone to access and use, so you can go to the website and apply to access the data should you be interested in doing so. So here I've just summarised some of the data we have on the cohorts. The mothers recruited during pregnancy, their children were followed intensively in collecting data on a range of health, demographic, environmental exposures, behavioural factors, development, education, parental psychological well-being. A vast array of data are available. And recently we were awarded funding to profile genome-wide methylation in a subset of the cohort. So we have genome-wide methylation data generated using the Infinium methylation 450 bead chip array. And so we've generated this on 1,000 mother-child pairs, on mums when they were pregnant, and the same mothers about 17 years later, on the children when they were born, when the children were age 7, and when the children were aged between 15 and 17. So we have this unique opportunity to look longitudinally across the life course, as well as intergenerational influences on DNA methylation. So the work that I'm going to talk about today focuses very much on this recent study, in which we were interested in whether gestational weight gain and or maternal pre-pregnancy body mass index were mediating the effect through altered methylation on birth weight, childhood adiposity, and later adiposity in adolescence. These are some data generated from an Epigenome-wide association study addressing that question. So at the top here we have a Manhattan plot which indicates the statistically significant observations this is cord blood methylation in children or offspring of mother at birth in relation to maternal underweight, in relation to maternal overweight during pregnancy, and in relation to maternal obesity during pregnancy. So we see a number of genome-wide association studies, or epigenome-wide association hits in relation to underweight. We see very little in the overweight category, and again we see when mothers are particularly obese, we're seeing some signals in relation to offspring DNA methylation at birth. So, as has been pointed out by various speakers this morning, can we really tell whether these associations are causal or not? And so some of the tools that we have at our disposal can be implemented here in terms of strengthening causal inference. And I'm just going to talk very briefly through some of these, and particularly focus on our favourite approach, and that being Mendelian randomization. So in terms of what tools, what methods can we bring to bear to try and understand causality, obviously randomized control trials are a gold standard, but often either implausible or extremely expensive. There are randomized control trials of nutritional interventions with epigenetic components to them underway, but in the main they're not the most obvious choice in terms of trying to interrogate causal pathways in epigenetic studies at the present time. So in a randomized control trial, the exposure is randomized, and you compare the risk of outcome. In a complementary approach, which we haven't used in epigenetic studies to date, but we have used in observational epidemiology to address the question of whether breastfeeding has an impact on childhood adiposity. And this is where we use two separate, quite independent cohorts with different confounding structures, and we compare the magnitude of association in those two different cohorts. And this is some very nice work by Marie-Jo Brion, where we compared breastfeeding in relation to childhood obesity and IQ in the ALSPAC study in Bristol and the Pelotas cohort in Brazil that has already been mentioned this morning. And we see consistency in evidence in relation to childhood IQ being higher in children who were breastfed, but not childhood adiposity. So it adds weight to or strengthens causal inference in terms of the interpretation of data. So potentially useful for epigenetic studies, but as yet has not been applied. Another method that we have used in relation to maternal overweight and offspring methylation is the use of a negative control design. And this is where you take a second set of data that has shared confounders and ask the question, do you see the same relationship with regard to your negative control exposure and your offspring outcome? And you compare the magnitude of association. So here we're postulating that our maternal overweight is altering cord blood DNA methylation through an in utero biological event. If that truly is an utero biological event, then you would not expect to see the same association with paternal BMI and offspring phenotype. If the relationship was confounded by some other factor, then you would expect to see these two associations of roughly equal magnitude. So here we have done exactly that. We've taken the top differentially methylated sites from our maternal overweight or parental overweight association shown a few slides ago. The maternal overweight and offspring methylation. And those are the plots shown in red here. So these are loci that are hypermethylated in children born to mums who are overweight, and these are hypomethylated, so hyper and hypomethylated. And when we do the same comparison in relation to paternal BMI, we see no significant associations. So this is just one of a suite of methods that strengthens causal inference that indeed we are seeing or can infer or conclude that the effect of maternal overweight on offspring methylation is a biological in utero event. So apologies to Stephen that dads don't matter in this case, but this is methylation, not microRNAs. Another study design that can be used and has been used, not by ourselves, but there are examples in the literature, is that of a sibling control. So where you have an exposure in the first pregnancy, and but the exposure is absent in the second pregnancy, and this overcomes the issue of familial confounding. And you compare the risk between siblings. And a nice example of that is the comparison of adiposity or overweight in children born to mothers who have had bariatric surgery or have been pregnant and obese in one pregnancy and have had bariatric surgery and had normalized weight in the second pregnancy. Clearly, it would be of interest to undertake epigenetic studies in this context. We've already heard today about the importance or the potential for incorporating longitudinal modeling in DNA methylation analysis, and this again can help to strengthen causal inference. One of the obviously a Bradford Hill criteria for causality being ensuring the temporal association that your exposure occurs before your the evolution of the phenotype or outcome, but that can be quite tricky in many contexts when we're thinking about epigenetic studies. The thing to note about longitudinal modelling of epigenetic patterns, though, is that although a temporal association in a longitudinal study can help overcome the issue of reverse causation, it may not overcome the issue of confounding. Your DNA methylation may be equally confounded in adulthood by socioeconomic status and confounded by the same factor, socioeconomic status, at birth. So longitudinal modelling or longitudinal approaches don't necessarily overcome all of the limitations that we're trying to circumvent. These are some data from the EWAS study I showed earlier, and here we've tried to address the question of the persistence issue that I asked Rob about earlier. So we observe a difference in cord blood methylation in children. How does that differential methylation track over time? So we're able to do this because we've got DNA methylation data in the same individuals at birth, at age 7 and age 17. And this is implementing some multi-level modeling to look at how those changes track over time. And these are just a few examples of the loci where we see differences. And I think it's important to note that that one needs to look at both the exposed group and the unexposed group. So here, for example, we have a locus that at birth is highly methylated or more highly methylated than the unexposed group, and it attenuates over childhood and into adolescence. Whereas the unexposed group, the level of methylation is fairly static. But we see other situations where we also see changes, natural developmental changes perhaps, or other environmental influences coming into play. So the unexposed individual at birth or group of individuals also show changes over time. And I think it's important that that one needs to consider both the methylation trajectory of exposed groups in the context of the unexposed group as well. But this is proving a really useful method to interpret the changes that we're seeing at birth and how they track and relate to phenotype. So the last part of my talk discusses the use of the approach that was introduced earlier by Andrea, called Mendelian Randomization. And this, the principle of this approach is that when we have a modifiable exposure that may be difficult to measure or is subject to confounding or reverse causation, we use a proxy measure instead of the modifiable exposure to get a better estimate of the causal effect of this exposure. And we use, in a Mendelian randomization, in an econometrics framework, it's called instrumental variables analysis. In this context, it's called Mendelian randomization because we use a genetic proxy or a genetic instrumental variable as a surrogate measure instead of our modifiable exposure. And because our genotype isn't subject to reverse causation, can't largely be confounded, it gives us a much better, much more secure causal anchor and allows us to make inferences about our exposure with regard to our outcome. So here we can use an allele score generated from genes or variants known to influence maternal BMI, and we can look at their relationship with child's DNA methylation. So we use these as a proxy for our measure of maternal BMI. Oops. And we can apply this to epigenetics. Apologies because it gets rather complex, but we can also do it in a second step where we want to know whether our child's DNA methylation is causally related to the evolution of childhood differences in BMI. So to do that, we need a genetic proxy for this measure, and we can do that because we know we can identify genetic variants that are very strongly correlated with site-specific DNA methylation. So I'm going to work through an example here of some analysis we've done recently, and this is based on the paper again that Andrea mentioned earlier, the observed association between DNA methylation and body mass index. So although the authors of this paper actually did go as far as identifying a genetic proxy for their methylation site of interest, they didn't actually conduct a formal Mendelian randomization analysis, but the principle of using a genetic proxy as a causal anchor was included in the paper. So we've done this at a number of different levels, and I'm going to try and explain in this schematic here. We've taken our 1,000 mother-child pairs, the data, genome-wide methylation data have been generated on this, and we've focused on methylation sites in this HIF3 alpha gene reported in the paper here. And so we're interested in knowing whether mum's BMI is changing infants' cord blood methylation, so we can look at that observationally and through a Mendelian randomization approach. We want to know whether offspring methylation here at age 7 is causally linked to offspring BMI, and again at age 15 to 17. We can also look longitudinally and use the temporal nature of our data to ask whether does methylation at age 7 relate to age 17 body mass index, or does body mass index or increased adiposity in childhood, is that driving methylation later in life? So a number of different scenarios that we can test using our data set. So the first step in that analysis is to use this is the Observational Association. We have maternal body mass index, and that is observationally linked to methylation at this gene in the child. So we use our maternal BMI allele score, and this has been generated with the new 97 SNPs that have been recently published through the GIANT consortium, which have been robustly associated with body mass index. So you generate a weighted score using 97 BMI-related SNPs, and you look at the relationship between those SNPs and childhood methylation. And so you test for the difference between the observed, which is this, and the expected estimates, which are B times C. And essentially what we see is that, the evidence that we're able to generate strengthens causal inference that maternal body mass index is having a causal effect on childhood methylation. And then we go to the second step and implement the same approach, but here we use a local SNP to our methylation site that proxies for our methylation site, and to ask the question, is methylation driving BMI? Is early methylation in the child driving subsequent body mass index? And the same principle applies. And here we conclude that there's no strong evidence that DNA methylation is having a causal effect in the child. And thirdly, to work the method through, we have this is whether child's own BMI is changing methylation later in the child's life course, and we see some evidence that that is the case. So I'd be happy to discuss the methodology and the intricacies of it with anyone who wanted it, but the intention was just here really just to give an overview of the method. So in terms of addressing the question, how important is maternal body mass index? Does it change offspring methylation at birth? And does it subsequently have an effect on childhood adiposity using this Mendelian randomization approach? We postulated at the outset that maternal body mass index alters cord blood or child's methylation at this locus, and that goes on to alter child's BMI. But having conducted the Mendelian randomization analyses, we conclude that the likely direction of causality is that it's actually the predominant effect is maternal body mass index acting through having some causal effect, direct causal effect on child's BMI, but not being mediated by early life changes in methylation. And maternal BMI is subsequently linked to causally linked to child's DNA methylation, but child's DNA methylation is not impacting on childhood BMI. So it corroborates the conclusion that the original paper made, that this association, which although is observed between DNA methylation and body mass index in adults, it's likely to be the changes in body mass index or adiposity that are driving the changes in methylation rather than vice versa. So I started by talking about this stepwise process in terms of establishing epigenetics as a potential causal mechanism. Firstly, revisiting those steps, how can we then improve each of the steps or improve the evidence or the tools and the data we have to address those particular steps? So we need better observational evidence using more refined measures of maternal exposure to really get a better description of what components of maternal overweight or obesity and maternal underweight are influencing offspring adiposity. We need improved technology for assessment of genome-wide DNA methylation. All of the data that I presented today is heavily reliant on the Illumina array, which has obviously limitations, to try and understand the connection between DNA methylation and subsequent childhood adiposity requires both of the above. And to really understand the causal nature, or whether epigenetics is mediating these associations between maternal overnutrition and offspring adiposity, we need increased awareness of the pitfalls of association studies and approaches for strengthening causal inference, and more widespread implementation of the what I've called the triangulation of evidence. So not relying on only one study design, but implementing a number of different tools to try and weigh up the evidence base for causality. And I'll just finish by acknowledging particularly Rebecca, Gemma and Mary in my group who did most of the work that I've talked about today. Thank you.